the webinars have allowed us to bring in people who would not normally come to our conferences because of distance. But we feel that especially one of the core purpose of SASSB, growing a community, getting students to connect with more mature researchers, getting people from different institutions and different organisms to be in one venue is something we're missing. So that will be part of the AGM. But without taking too much time, I would welcome Finance to introduce the speaker and look forward to, I've got my pen, so I'm taking my notes and getting initiated. Thank you.
Well, thank you very much for that, Mutama. Yeah, it's a pleasure for me to introduce Marike Palmer. At least for this audience, she should not be too unfamiliar unless they recently joined this community, because we were talking about it. Marike presented her honours work at for the first time at a conference of the SASSB in Arniston in 2012. So we're now 10 years later. And I must say, from that honours presentation, and she won the prize with that presentation. So we were very happy that time, but she's really gone from strength to strength. She's finished a PhD here at UP, then did a short postdoc here, and then went over to University of Nevada at Las Vegas. So she's had the opportunity to make lots of money for the last nearly 3.5 years. I don't know how successful she was visiting is it called the Strip? I think.
Yes, unfortunately, I haven't had any luck visiting the Strip.
Yeah, well, I think it's probably a sign of a good scientist if you know that you should stay away from the Strip. But yeah, so she's always been active in taxonomy, and that has also resulted in her being drawn into the SeqCode activities, and she will tell us today about it. So we're always happy that there are some younger faces in a committee like this, and we're lucky we have a number of them in our group. So we're very happy with that. So over to you, Marike.
Thank you, Faunus, for the introduction. Let me just share my screen. Let's do that. Okay, so it is really a great pleasure to be talking to you today as an SASSB crowd. As Faunus said, my first ever conference presentation was in Arniston in 2012. And since then, even though my work has changed quite a bit, it hasn't strayed far from taxonomy and systematics. And it really is a great pleasure to be talking to you today. So today I'll be talking to you about the SeqCode specifically, and I am currently a postdoc scholar at the School of Life Sciences at the University of Nevada, Las Vegas, as Faunus said, but I'll be talking to you as one of the steering committee members of the SeqCode specifically. So I don't think as an SASSB audience, so let me first start off by actually acknowledging the rest of the SeqCode steering committee and our funding sources. And you can actually find us on Twitter at the handle seq_code. And also additional information can be found on the ISME website. And I would like to particularly thank Masha, Brian, Phil, and Miguel, who spoke to the SASSB webinar audience in May of this year, and Ian and Barney for allowing me to actually use some of their slides for this talk. So let's jump in. I don't think as an SASSB audience, I need to convince anyone that nomenclature is important and that it is definitely more than just biological stamp collecting. But for anyone that might still need a little bit of convincing, I think one of the best ways of describing nomenclature was by McGee in 1993. And that was described as being the working technical language of microbiology. And I actually think this is true for all of biology. So basically, nomenclature forms the language that enables us to effectively communicate our science to one another. And as with all languages, we have a set of rules that we need to follow to ensure consistency of that language. So the codes of nomenclature can basically be seen as the sets of rules that govern the naming of the taxa. So this is not for delineation or classification purposes of the taxa. It's specifically focused on the actual naming of the taxa. And the three main goals probably of these sets of rules are firstly to ensure precise names of the taxa. So in this case, we have precise names that are associated with specific taxa that ensures that we can use those precise names when we're talking to one another and that we're consistent in terms of what we're referring to when using those names. The second goal would be to basically ensure that each taxon only has one valid and correct name. So as a matter of taxonomic opinion, sometimes this might differ in the usage of the names that are being used by some people. So they might be using older or different valid names for a taxon, but usually we consider only one valid name as being the correct name for that taxon. And the third point would be that we basically ensure that each name refers to only a single taxon. So the second and third points might be a little bit intertwined, but basically with the second point, when we're saying a specific name, we want to make sure that the taxon only has one correct name, whereas the third point, when we're saying a specific name, we want to make sure that only refers to one biological entity that we can recognize instead of referring to numerous different ones. So the way that these sets of rules are basically set up is by linking specific names, the precise names that we're using, to nomenclatural types. And the types are basically the things that we use to determine whether we have sufficient similarity between our taxon and that nomenclatural type to actually give them the same name. And if they're sufficiently dissimilar, it actually warrants the creation of a new name. Another very important principle in codes of nomenclature is priority. And this is basically the practice of ensuring that the first published valid and correct name for a taxon is respected. So this kind of ensures that we use the first valid and correct name for a taxon throughout the history of that taxon. And even though taxonomic opinions might change, that first name would always be the one that's being respected. So if we're thinking about linking names to types, in the Shenzhen code, we have this description of a nomenclatural type as the element to which the name of a taxon is permanently attached. So this serves as an anchor point for a specific name, which actually allows us to use that stable, probably in quotation marks, names to build our taxonomies on. And if we're looking at the different types of nomenclatural types, we can actually look at the ICN, so the International Code of Nomenclature for Algae, Fungi, and Plants. And again, this would be based on the Shenzhen code. We basically have herbarium specimens and illustrations and also viable cultures for yeasts and algae that can actually serve as nomenclatural types. And similarly, we also have various different types of nomenclatural types in the International Code of Zoological Nomenclature, so the ICZN. So in this case, we can have microscope slides, or we can have fossilized parts of animals, or full animal fossils, or actual animals, preserved animal specimens, or parts of animals that can serve as nomenclatural type under this code. And this actually allows of data that is appropriate for the different taxonomists to actually describe and name their taxa. In contrast to this, the International Code of Nomenclature for Prokaryotes the ICNP, which would be how I refer to it throughout the talk further, the ICNP, actually only allows viable pure cultures of microorganisms that has been deposited in 2 independent culture collections on different continents to serve as a nomenclatural type. So to just summarize this, we can see that the ICN and the ICZN all allow various different types of nomenclatural types within those codes. However, since 2001, the ICNP only allows viable designated strains as like from cultures, so pure cultures grown in the laboratory and deposited in culture collections that are viable to serve as a nomenclatural type. So just as a thought experiment, kind of, if we were to consider this restriction or this requirement for nomenclatural types in any of the other codes, this would be the equivalent of embryos or cryopreserved mating pairs or actual sexually reproductive individuals from a population that needs to be preserved as nomenclatural types going forward. So obviously we know that if that was the case, none of the dinosaurs would have been able to be classified. There would be a plethora of orchids and other plants and even mycorrhizal fungi that would not be named under those codes. So in this paper, we argue that the ICNP is really unique in the fact that it's only accepting of a single type of nomenclatural type. And we also argue in this paper that this has actually hindered the progress of prokaryotic taxonomy. So if we just compare in terms of valid names across the different kingdoms of life and also including the viruses, we can actually see that for the archaea and bacteria, we only have about 20,000 validly published names for these, whereas all of the other cellular organisms far surpass this number in terms of the species that has been described in those groups. So some people might say that it's too harsh for us to blame the restrictions of the ICNP for this lag in progress. But if we actually break this down, can we actually consider these restrictions as to blame for this lag? So over here, I have a basic outline that we as prokaryotic taxonomists have typically used for describing new species. So this typically starts off with obtaining isolates and pure cultures of those isolates. Then we continue by doing chemotaxonomic and phenotypic characterization of those strains in the lab. And this is typically followed by looking at phylogenetic cohesion by looking at specific marker genes. So we would do marker gene sequencing, and 16S ribosomal RNA has, of course, been the genes for those have, of course, been a really staple in taxonomy previously, but we have moved more towards several different marker genes being sequenced. And then ultimately the last step in order to get the names validated would be to submit these cultures to culture collections. And then in the case of it being published in the International Journal of Systematic and Evolutionary Microbiology, we would get validation of those names. Or if this was published outside of IJACM, we would request validation to occur on a validation list, and this ultimately leads to validation of the names. However, as genome sequencing has become much more frequent and cheaper, we're at a position where these type of characterizations are actually largely being replaced by genomic analyses. So what I mean by this is where I've actually shown this figure several times at an SASSB talk, and this is basically the global overview map of the Kyoto Encyclopedia of Genes and Genomes for metabolism. So the global overview metabolic map, and this basically indicates all of the reactions with the dots being specific compounds and the reactions being the lines that connect these dots, and how metabolism is kind of intertwined across the board of organisms. And we're really in a position that we can kind of predict the metabolism of various different organisms already. And this is really routinely used for bacteria and archaea. And not only can we predict the phenotypes of those microbes from this data, we're really also at a point that we can generate hypotheses of trying to cultivate microbes or actually design experiments for testing specific physiologies of interest. And then, as I said, we typically use marker gene sequences, or traditionally the 16S rRNA gene, to actually investigate phylogenetic cohesion of taxa, but we're really at a point now that we can use phylogenomics to do that. So in this example, we have Geobacillus species, and indicated in boldface would be the nomenclatural types for those species. So in this group here at the top, where I would like to focus your attention, is the Geobacillus stereothermophilus type strain. We also have a Flavobacterium thermophilum type strain in there, which is clearly incorrectly classified, but we also have the type strain for Geobacillus luteanicus in there. So using genomes, we can ask the question whether this actually represents a single species, whether this represents two species, or potentially more because of this Flavobacterium thermophilum type strain in there as well. So the fact that we have genome sequences available to us rather than just a small set of marker gene sequences means that we can really investigate the cohesion among individual gene trees. So we can look at all of the individual genealogies for the shared gene content of these taxa, and we can overlay those trees on top of one another and look for the points of congruence to actually identify species boundaries. So in this way, we can really much more robustly than ever before identify this entire group as a single cohesive species, even though there might be some more or less genomic similarity across the board for these. So this is already something that we're doing for bacterial and archaeal isolates. So as I said, we're already replacing a lot of these characterization steps with genomic analyses. But this isn't the only bottleneck or problem when we're looking at this typical flow of prokaryotic species description. The largest bottleneck is actually at the point of isolating and obtaining pure cultures of these organisms. So we're already at a point that we know that the vast majority of the microbial diversity out there is not yet culturable. And oftentimes, if we do obtain stable co-cultures, we're unable to get them, those specific organisms within that community, as pure cultures, because they might be obligately dependent on community members with them. So this is a slide that I stole from Ian Sutcliffe, and he always talks about the sublime scale of the microbial world, so the diversity in the microbial world. And this is one of the more recent estimates of how many taxa we think might exist in nature in terms of bacteria and archaea. And the current estimate is that maybe around 10 million species of archaea and bacteria exist. And based on the actual names that have been validly published for bacteria and archaea under the ICNP, we're at a point that we have around 10,000 names being validly published, which makes up 0.2% of all of the bacterial diversity that we currently think might exist. So on this table over here, if the entire red spectrum makes up what we think currently is microbial diversity in general, we're really only at a point that we've somewhat started describing this first block in this entire array. So you might ask, how do we know how many taxa actually exist in nature, and how do we know this diversity exists? And the answer to that would be that we're in a position to use environmental genomics. So just as a quick rundown, basically we can take an environmental sample that contains a specific community of organisms, In our case, specifically, we're looking at bacteria and archaea, and we can bulk extract the DNA within those samples and sequence all of that DNA. We can then follow this up by actually binning these sequences into individual bins after the fact. So we're basically using different algorithms to assign the sequences back to what we consider the specific populations within the sample initially was. So in this way, we can actually obtain population genomes for the different populations within a community of an environmental sample. And when we actually apply this, we can use the exact same methods for species delineation and circumscription for these environmental genomes as we're currently using for the isolate genomes. So just as an example, we have this genus Geocranenicus, and this is in a paper that we recently published in Nature Communications. And in this genus, we haven't got any cultivated members yet, but we have numerous different metagenome assembled genomes, so MAGs, and all of these genomes come from metagenomic sampling. And it's a group of thermophilic archaea, so they live in geothermal springs. And we have various different genomes from the geothermal fields in Tengchang, China, and also from Yellowstone National Park. And we name these taxa Geocranenicus arthurdei for Arthur Day, who played a pivotal role in establishing Yellowstone National Park, and Geocranenicus wangii and dongyi for Chinese scientists that have played a vital role in environmental microbiology. So if we apply the same principles that I just discussed for the Geobacter example, we can also look at these individual gene trees constructed for all three of these taxa, for all of the genes that they end up sharing. So the marker genes that gets used in a phylogenomic analysis, we can overlay these, and we very clearly still see these very cohesive and distinct groupings occurring that kind of are congruent with our species hypotheses that we've generated. So we can really use the exact same methods that we're currently using for isolates. We can use that already for environmental genomes. And we still get these really robust species identifications. So if we're looking at, in terms of the diversity that we then know exists currently, we can use this total bar specifically to look at closer, but basically of all of the diversity that we know exists, and again, these numbers might be a little outdated already, but this was the estimates in 2018. So we're literally at a point that we're increasing our known diversity daily, virtually, with sequencing of more metagenomes. But if we're looking at all of the taxa, we have a tiny fraction of that. So we have 18% of all of the diversity that we know exists already that have validated names for those specific taxa. There's another almost 20%, so maybe 15% that would have proposed names, but these names are not valid and they cannot be validated under the ICMP due to the restriction of only allowing viable cultures to serve, pure cultures to serve as nomenclatural type. And then there is a massive majority that only has placeholder names, which would typically be alphanumeric identifiers and which makes communication incredibly difficult regarding these taxa. So to address this, in 2016 already, Barney Whitman wrote this proposal to actually expand type material so that the nomenclatural types that are being allowed under the ICMP, to expand that to actually also allow DNA sequence data. And since then, there has been various papers that came out specifically arguing that we need to incorporate or accommodate the uncultivated diversity that we know exists in some or other nomenclatural code. We need a way to effectively name and validate these names and ensure priority of these names to facilitate communication about these taxa. And in 2020, this roadmap paper as a consensus statement was published in Nature Microbiology with various names that you might recognize on here, and also some very prominent people that are involved and actively doing biodiversity exploration that includes the uncultivated taxa. So in this paper, we had this specific roadmap outlined in terms of what the routes forward might be. And although this is a lot of text and I don't expect you to read this, the take-home messages that we have from here was that we had Plan A as being a mandation of the ICNP to accept DNA as nomenclatural types, whereas Plan B was to develop a new code of nomenclature that would use genome sequence data as nomenclatural types. And while this paper was still finishing through its editorial process, the SASSB, which is the governing body of the ICNP, actually voted to reject this proposal by Barney Whitman. So at that point, it was completely inevitable that we had to follow this Plan B route as Plan A has been rejected. So since then, we've been busy with first off establishing an informal SeqCode organizing committee, and we've basically prepared a first draft of the SeqCode in 2020, which was under the umbrella of ISME, so the International Society for Microbial Ecology, again, which several of you are involved in. And in 2021, based on funding obtained by Brian Headland and Anna Louise Reisenbach, they actually that funded this work. A series of online workshops was actually held in February 2021, where almost 1,000 registrants from more than 40 countries actually attended these workshops. And the informal SeqCode committee was very much encouraged to continue with the development of this independent code of nomenclature. So in 2022, so this year, there was a roundtable discussion specifically on the SeqCode at ISME 18, and we also published the SeqCode version 1.0 just a couple of months, 2 months ago. So this paper in Systematic and Applied Microbiology describes the development of the SeqCode, and this was shortly followed by the actual publication of the SeqCode itself, and then guides on how to use the SeqCode. And from here, we decided to obviously make the SeqCode as user-friendly and as useful as possible, and to ensure that we have a forward-looking perspective on this, we decided as the SeqCode Steering Committee to have an online platform where we can have this name registration and validation occurring. So the actual data validation and the actual list of validated names is housed electronically and not a discrete set of rules printed somewhere that only gets updated infrequently. So for that purpose, we want to have the SeqCode be a living document that is community driven, but we also want an interoperable, useful registry system to do this. So currently the SeqCode registry is still in beta testing phase, but it is open for submission, and we have several names that have already been validated under the SeqCode. So for anyone that's interested in checking this out, this would be where the SeqCode registry is available. And just as a short overview of how this process of registering names work, basically anyone with an interest in taxonomy, microbial taxonomy and systematics can apply for contributor status within the SeqCode registry. And then as a contributor, you can then create the name for your taxon within the registry itself, which generates the pages for species and any associated lineages that you might need. And then you as contributor would fill out all of the QC and metadata associated with the pages for those taxa. And then it goes through a nomenclature step, nomenclature review step, where the actual nomenclature for the names gets reviewed. And if the name gets approved, we can continue, where the curators would do a data quality check. So we need to use high quality genomes for this purpose. And if any of the genomes fail the specific QC checks, we as curators would then evaluate the explanation for those failed checks and determine whether that genome would still be suitable and a viable nomenclatural type for that name specifically. So if at that point, all of the nomenclature and QC checks were passed, we basically obtain approved registry lists. And then upon effective publication in any journal, so not a specific journal in contrast to the ICMP, in any peer-reviewed journal, that publication would, the effective publication would trigger validation of that specific registry list. And at that point, when the list becomes validated, we have the date of priority for that name. So in the SeqCode paper itself, we have this flow diagram of how you can physically go about doing this. And when we start off with having a new name for our new taxon, again, we're not governing how species delineation is occurring. It's specifically focused on the naming of those organisms. So if you have sufficient evidence that warrants the creation of a new name, we have several different paths through which this registration and validation can occur. Hopefully in the future, we would have a partner journal through which the actual peer review and SeqCode registration would occur simultaneously, and that it's an integrated system. But this would be something that we'll be pursuing in the future. But currently we have this path 1 and path 2 routes that you can follow to actually validate your names. So under path 1, which would be the far more encouraged route to follow, you would be generating and registering the name during manuscript preparation, and the actual SeqCode evaluation validation process, so approval of your name, occurs simultaneously with independent peer review in order to catch any potential errors, so any orthographic errors or anything that the curators might suggest might need improvement or slight correction of. And then at the proof stage of your manuscript, you can incorporate those different recommendations from the curator team. And then upon publication, you would actually get the date of priority when the DOI gets added to the registry specifically. And you would end up with a validly published name. Of course, there are various names. As I showed you, there are various proposed names that can't be validated under the ICMP. And in order to accommodate those, we have this path 2, where we would already have existing publications that includes the name. So the minimal requirement for the effective publications is that the name has to be in that paper. And from that point, we can basically do the SeqCode registration with the already existing effective publication. And if all of the necessary QC checks are passed, then the name would actually become validated in the SeqCode registry, and that would be the date of priority. So if we look at what the next steps for the SeqCode would be, I told you that the SeqCode version 1.0 is already published now, and we very much want this to be a living document that serves the community optimally. So the next steps would be to actually establish a community-driven SeqCode committee, and this would be through the adoption of the formal governance processes, which we call the statutes. And basically, the SeqCode committee will consist of the larger SeqCode community, who would be anyone that is involved in the SeqCode itself. So anyone that is interested in microbial taxonomy or systematics or nomenclature can join the SeqCode community, and they would become voting members by signing up to the SeqCode community. And then this committee consists of 5 administrative components. So we have the executive board, the legislative commission, the Reconciliation Commission, then the Registry and Nomenclatural Working Group, and lastly, the Standards Working Group. Because as I mentioned, we really need to make sure that we use the best possible genomes that we can as nomenclatural types. And for that purpose, we're actually now at a point that our standards are very strict in terms of what can serve as nomenclatural types, but in the future, it can be evaluated based on the data and the uptake of the community that is available at that point. So we've already adopted the statutes version 1.0 as it is currently, and I would encourage anyone that's interested to please sign up and participate in this initiative. And with that, I would like to end and say thank you for your time. Thank you, Faunus, for the invitation. And anyone that is interested, please check out, this is our Twitter handle again, the SeqCode information on the ISME website. And if you're interested in joining the SeqCode community, it would be this QR code, join. And if you're interested in the governance structures or taking up any governance roles within that administrative components, this would be the QR code to sign up for that. And I'd like to end off with a quote that kind of, I feel, embodies the spirit that I hope everyone on the SeqCode steering committee and anyone that is willing to join the SeqCode community as a whole can kind of embrace. And that is that the difficulty lies not in the new ideas, but rather in escaping from the old ones. So thank you for your time. I'll leave this up for the moment, and you're welcome to take photos, or I can also make the presentation available at the end. Thank you.
Thank you very much, Marike. Of course, for me, it was an enjoyable talk. It's always nice to hear this and sort of reflect on where we came from and what we've done. We're now ready. If there are any specific questions, just raise your hand, indicating that you want to ask a question, or you can put a question in the chat box if you want to do that. But you are welcome to now join in the discussion. So any specific questions? Gemma, you can unmute and
Hi, can you hear me?
Hi, yes.
Hi, I have just got a question about well, just the process or the idea of using sequences for species identification. So I don't work on microbes, but I'm just starting a PhD and aiming to work on fungi. But in my understanding is that in fungi, the fungal world, there's also the same problem of a very small proportion of them are culturable. And so at least in South Africa, we're quite limited to only being able to name the ones that are culturable. And I just wondered what you think about the application of the similar techniques you're using to other taxonomic groups like fungi.
So, of course, having much smaller genomes for most of the prokaryotes makes it vastly easier to develop and fine-tune specific tools that will allow you to investigate cohesion of the taxa specifically than it might be for the eukaryotes. But I do think that so even though the SeqCode doesn't govern how species circumscription should occur, I think all of us have our own taxonomic opinions on what would be the best approaches to do that. But ultimately, I think as genome sequencing continues to decrease in price and increase in its regularity of being used, I don't think there would be reasons to not include DNA as nomenclatural types for other taxonomic codes, or other nomenclatural codes going forward. But it really is a point. So hopefully the SeqCode initiative can sort of serve as a way to do that going forward, or perhaps how not to do it potentially as well, in the way of actually incorporating DNA into existing codes. I think from the SeqCode steering committee's perspective, I think all of us would have preferred inclusion of DNA in the ICNP itself. But I do think that, at least for the prokaryotes and the archaea currently, genomic-based species delineation is turning into the gold standard, if not already.
Okay. Awesome. Yeah, I mean, that makes sense. But yeah, I'm excited to see where things go.
Yes, hopefully it's as exciting as I think it might be.
Yeah. Fingers crossed. Okay, thanks, Mary.
Thanks.
Diego?
Sure, thanks. Thanks for the great talk. It was very interesting. I'm just wondering from a practical point of view, say now someone is writing a paper using MAGs or SAGs, should we just still use GTDBTK, for example, to speak about our MAGs and SAGs, and then eventually we'll use specific names that come from SeqCode? Is that the idea, more or less?
So the idea is that if you're doing species delineation and circumscription, you can do that in whatever way is appropriate. And I am a big supporter of the GTDB. So GTDBTK is an absolutely perfect way of doing that as well, using various lines of evidence, including phylogenies. And the idea would then be for you as the taxonomist that is working on the group that you probably know best, to have that species delineation step incorporated into your paper, preferably, and actually provide the names for those taxa. So you would basically, as a contributor to the SeqCode registry, you would have the manuscript in preparation, for example, and actually already register the names that you want to assign to your taxa in the SeqCode registry so that it can go through the different evaluation processes of the names themselves. And upon publication of your manuscript describing the taxonomy or the physiology of the organisms or whatever, at that point, we would hopefully be at a point to be able to validate the names if the genomes that are being used, so the MAGs and SAGs, are of sufficient quality.
Awesome, thank you so much. So that would be more for like novel genomes that we find. But in terms of the existing ones,
so
like
we
phylo, like, I mean, with archaea, like, I don't know, so many, we've changed, for example, in the manuscript so many times from Crinoarchaeota to Thaumarchaeota to Thermoplasmatota, now like it's just messy. So what would you recommend to do in that regard?
So it definitely is somewhat difficult, and as all of us that are involved in the SeqCode actually consider ourselves involved in taxonomy as well, it's hard to not like obfuscate the nomenclature by looking at taxonomy specifically. But in terms of the SeqCode itself, we don't specify any particular taxonomy. So any code of nomenclature shouldn't be used as a means to prescribe taxonomy to people or to prescribe taxonomic opinion to people, but rather serves as a set of rules to name those organisms. So where I might be convinced your taxon could be a class or a new phylum, ultimately peer review through the actual publication process is going to be what decides whether your rank that you want to describe is warranted or not. So if the reviewers to your manuscript still decide that no, this taxonomic opinion that you're expressing isn't robust enough, or they don't agree with it, the actual peer reviewers might still ask you to do changes, but ultimately what is published ends up becoming the validated record of the names in the registry itself based on the actual peer-reviewed publications. Does that make sense?
Yeah, perfect. Thank you so much.
Sure.
Yeah, and I think over time, what we will have is that many of these names will filter into the GDTB as well. The moment they become valid, those names can be incorporated. The next question is from Nicola.
Yes, hi, thanks. Thanks, Marike, for a great talk. That was really super interesting and very informative, actually. I learned a lot about prokaryote nomenclature, actually. So I've got two questions, actually. Well, maybe one first is maybe quite a small one. I think, sorry, maybe I just missed this because I missed the beginning of the talk. But do you require a full genome as the type, or can it just be part of the genome?
So currently, I didn't actually go into the QC requirements or anything. So it's a completely good question. So currently, of course, we have various different algorithms and approaches for estimating completeness and contamination of these environmental genomes. So currently the standards are quite strict. So we basically ask for high quality draft genome. So it needs to be at least 90% completeness, less than 5% contamination in terms of estimates being used. So it's technically not necessarily the full genome, but of course, if we do get a full genome that's assembled to replicon level, that's absolutely fantastic. And then one of the very strong recommendations would be that it does actually have a 16S, or basically the entire ribosomal RNA operon would be a very strong recommendation. But we do appreciate that sometimes with these binning algorithms, it's very, very difficult to actually robustly identify 16S sequences, so the scaffolds that contains the actual ribosomal RNA operons, to bin that accurately to the rest of the metagenome assembled genomes. And in those cases, everyone on the steering committee does not agree with this. But my personal view is that it's probably better to not have a 16S than to have the incorrect 16S associated with a particular name. So in those cases, we would rather accept a genome as a nomenclatural type that doesn't contain a 16S than forcing people to fish for 16Ss that might be the incorrect 16S. Potentially depending on, so our forward-facing vision ultimately would be that there would be specific working groups that might work with specific taxa of interest. So for example, if there's intracellular parasites, like the chlamydia, hopefully we would be in a position to have a chlamydia working group, and they would ultimately, based on the community opinion and input, they would be able to propose changes to those standards that we're currently applying across the board. So potentially if we have something that is very difficult to obtain full genomes for, we could potentially use parts of genomes as the nomenclatural types, as long as we're able to confidently and robustly identify those taxa using that piece of DNA. But we're not there at the moment yet. So at the moment, we're still at a point that we're encouraging and supporting very high standards for the genomes being used.
Okay, thank you.
Sure.
Yeah, so then if I may ask my second question, I just thought, what an amazing example of cooperation and, probably international teamwork. I think it's such a great way maybe also to teach nomenclature to students, actually, this whole process. But I'd also wondered, I've got colleagues who I think sit on the plant committees and all, and it must be so difficult to actually working with the rules. And I wondered if there's any lessons or things that you've learned from the existing, the plant and fungal codes and the zoo codes that you were able to sort of, know not to make that mistake when you're putting these set of rules together.
So that is something that's quite a task. But I think for me personally, at least, I've come to accept that we definitely do need rules. As I said, this is a language, the language that we use to effectively communicate with one another, and any language requires some rules associated with it. And we have, while developing the SeqCode and actually working on the SeqCode document itself, the code itself, tried to simplify the entire code in comparison to the ICNP itself. So I do think that probably one of the largest lessons that we learned from that is that we need to try and avoid ambiguity as much as possible. And I think the fact that we're pretty diverse in terms of languages, in terms of scientific background, group that actually work together, we actually, so oftentimes 6 or 7 of us would read that specific rule and we would all be like, oh yeah, no, that's obvious. Obviously it needs to be that way. And then someone from a different background would be like, oh no, but this doesn't make sense. They understand this and this and this from that rule. So even though that actually made for quite a eventful collaboration across the board, I think that actually helped a lot with us trying to eliminate a lot of the ambiguity that was currently in the ICMP and hopefully not transfer that ambiguity over to the SeqCode itself. But yes, ultimately, I think even though we tried to simplify the SeqCode and have probably 30 or 40% fewer rules than the ICMP initially, it is somewhat inevitable that we do have various different rules and recommendations accompanying those rules. So some people would argue that there's still too many rules incorporated in the SeqCode itself, but I think like we've spent a lot of time evaluating each and every one of the rules that ended up in the SeqCode itself. And I think most of us kind of felt strongly that those were at least the minimal set of rules that we could unambiguously come away with. Faunus might have an opinion on this as well.
If I can just add here, one of the issues that we also had to deal with was that in future, we would like to have the bacterial codes joined. And therefore, we could not move too far away from the current code because we want to be able to merge with them if it's possible without too many trouble. So many of the rules were maintained because this is the way that it's been approached. So we were a bit pragmatic also in that. And just from my side, it was quite an interesting thing. We had meetings where it was 5 o'clock in the morning in Australia the next day. Somewhere in the middle of the day in America, and I was sitting at home at 10 or 11 in the evening, and we had these discussions. But it was quite interesting. There's another question in the chat, Marike, from John Osorio, and he says, what plan do you people have to obtain information from complex ecosystems where not many people like to go or where extremely difficult to get access? Looks like to obtain information from those areas is going to take so long. So is the plan to get more collaborators that work with extreme environments?
So I think as the SeqCode steering committee side of things, it's not like we would, so stuff that we are for the SeqCode specifically trying to obtain funding for would be development and improvement of the SeqCode registry and trying to take all of the current proposed names in literature and candidatus names to get those incorporated into the SeqCode itself where possible. But as the SeqCode committee as a whole, I don't think there would be massive drives towards sampling specifically extreme environments independently. But of course, we're all scientists and all of us are interested in biodiversity exploration. So aside from the SeqCode committee itself, I think all of us do actually pursue with strong collaborations, definitely. As Faunus also described collaborations on the salterns in the talk in May that Miguel gave. I do think independent of the SeqCode committee, probably all of us are pursuing trying to obtain better coverage of the biodiversity in all environments, including extreme environments. I don't know if that answers the question.
Yeah, if I can add, I think if you look at the SeqCode committee, there are a number of people that work in these environments, and they really had the drive to get names for these organisms that they've isolated, because clearly in that environment you often do not have pure cultures that you can name. So it's interesting that, yes, the committee also have quite a large representation of quite diverse environments from that. I think these are all the questions. Thank you very much for your time, Marike. It was a pleasure to listen to you. 10 years later, it's still exciting. And I must say to everybody, if you want to employ her, she is, what's the word, open for work or whatever. So she,
exactly, in the job market.
Job market, she would shortly be been there as a postdoc for 4 years and is now ready to move on. So spread the word. Here is a gem to employ. That's just me marketing my PhD student. So thank you.